Good morning, dear students. I'm Dr. P.THENMOZHI, Head of the Department of Zoology, St.Joseph's College of Arts and Science, Autonomous Cuddalore. Today I'm going to talk about plasma membrane. First of all, we can see what is plasma membrane. Plasma membrane is the outermost layer of all the animal cell. You know very well that the difference between animal cell and plant cell. Animal cell is made up of plasma membrane. Its thickness is about 5 to 8 nanometer. It is having a special character called a selective permeability. It serves as an outer boundary of the cell. It allows some substances to cross more easily than others. It is made up of phospholipids, proteins, and conjugated molecules. It can separate and protect the cells from external environment. It provides a connecting system between cells and its environment. It is also called as cell membrane. So we can define plasma membrane as it is an outermost envelope surrounding the cell that separates and protects the cell from the external environment and provides the connecting system between the cell and its environment is called as a plasma membrane. Now we can see the structure of the plasma membrane. The structure of the plasma membrane can be explained in many theories. Nearly 4 or 5 theories are regarding it. One among them is the most successful one. We can see one by one. The structure of the plasma membrane, it is composed of two layers of phospholipids which are connected back-to-back positions. The phospholipids are the lipids with a phosphate attached to them. Here you can see the phospholipid bilayer. It consists of two types, two ends. At one end it is having head, another one end it is having a tail. There are two layers of phospholipids. That's why it is called as phosphobilipid layer. And one is the polar, the two polar regions. Outermost region are called as the polar hydrophilic heads. It is having the head which can allow the water. But in the tail region, that is, it is facing, both the tails are facing in the middle. That is called as non-polar because it is not having head. So it is non-polar. It is a hydrophobic tails. That means it reject water. It is not loving water. Hydrophilic means it loves water, allow water. Hydrophobic means it doesn't allow water inside. This is the rejected theory, sandwich model of plasma membrane proposed by Daniel and Davison model. In the year 1935, cell membrane is a lipid bilayer sandwiched between two monomolecular protein layers, that is you can see in the diagram. Fluid mosaic model is a successful model proposed by S.J. Singer and G. Nicholson in the year 1972. This is called as fluid mosaic model. What is fluid mosaic model? Cell membrane is a lipid bilayer in which proteins are partially embedded like floating icebergs in the sea. The fluid mosaic model describes the plasma membrane as a flexible boundary of a cell. The phospholipids move within the membrane. You can see the lipid molecules are present in the fluid state capable of rotating and moving. The proteins occur as a mosaic of discontinuous particles that penetrate deeply into or even through the lipid sheet. The globular proteins are irregularly embedded in the lipid bilayer. There are two types of plasma membrane proteins. They are extrinsic proteins and intrinsic proteins, otherwise called as peripheral membrane proteins or integral membrane proteins. We can see one by one. Extrinsic membrane protein. What is extrinsic membrane protein? From the name itself, we can see that the proteins which are present in the outer side or both the sides, that is outer side and the innermost side, these protein molecules are present. That means the proteins are loosely associated with the membrane surface itself. both the inner surface and outer surface. It is located entirely outside of the lipid bilayer, that is either on the extracellular side or cytoplasmic surface. Cytoplasmic surface means inside the cell. It is also called as peripheral membrane proteins. For example, the cytochrome C is one of the peripheral protein which is present in the mitochondrial membrane. Then another one example is antigens. You can see the antigens, the cell surface, they can make identity so that it is called as identity markers. Intrinsic membrane proteins. What are intrinsic membrane proteins? Which are directly incorporated within the lipid bilayer. They tightly bound to the lipid bilayer. It provides channels for water-soluble substances, and it is also called as integral membrane proteins. For example, transmembrane proteins, transport proteins, channels, permeases, that means pumps. Here you can see the diagram which clearly illustrates the glycoprotein, extracellular fluid, glycolipids, then transmembrane proteins inside the cytoplasm layer, peripheral proteins, then filaments of cytoskeleton also. You can see the membrane is a collage of proteins and other molecules embedded in the fluid matrix of the lipid bilayer. Then you can see the chemical composition. Plasma membrane is composed of lipids, proteins and carbohydrates. The actual components differs from the tissues to tissues because it depends upon the type of tissue in which it is present. The lipids of the cell membranes are phospholipids, glycolipids, sterols and cholesterols. Membrane carbohydrates. What about the membrane carbohydrates in the plasma membrane? They play a key role in cell-to-cell recognition. That means they have the ability of a cell to distinguish one cell from the other. Example, different types of antigens, major histocompatibility complexes. Important in organ and tissue development, they are very, very important. They are the basis for rejection of foreign cells by immune system. Some of the plasma membrane also they can capable of rejecting the foreign cells which are causing diseases to our body. Movement across the cell membrane is shown in this picture. That is protein channels. You can see different types of protein channels in which through which the substances are transported. Then inside the cell and outside the cells, how does the different protein particles or other particles are sent in and outside the cell is shown in this picture. You can see the transport proteins, one which you can see in this diagram, the red colored one. That is the transport protein which is having middle, there is a pathway of allowing the needed substances or waste materials. It may be needed substance can be taken out outside the cell and transported inside the cytoplasm, or some waste materials from inside the cytoplasm can be exerted out through this channel. Okay, next important function is diffusion. Diffusion is one of the important function you can see in the plasma membrane. That is, some of the substances or some of the liquids can move from high to low concentrations. That is what called as passive transport. In passive transport, there is no much stress is taken part by this plasma membrane, so that no energy is needed. Here you can see the movement of water in the way of osmosis, simple diffusion. Osmosis means what? It is the diffusion of water across the selectively permeable membrane is called as osmosis. That is, regulating the water flow through the plasma membrane is an important factor in maintaining homeostasis within the cells. What is passive transport? When the cell uses no energy to move particles across the membrane, then it is called as passive transport. Here you can see the plasma membrane in which the two bipolar sites of the phospholipids, so that outside and inside of the cell there is a movement of flow, that is the concentration gradient. Through the concentration gradient, it diffuses inside. Passive transport by proteins. How does the proteins are doing passive transport? The passive transport of materials across the membrane using transport protein is called as facilitated diffusion, because it facilitates, the proteins are facilitating, they are helping the substances from outside to inside or from inside to outside to move. So here you can see the channel proteins in the plasma membrane. It moves, the fluid moves across the concentrated gradient. Facilitated diffusion. Diffusion through protein channels is otherwise called as facilitated, because it is facilitated by the protein channel. Channels move specific molecules across cell membrane, so no energy is needed. So here also you can see in the hydrophobic region of the protein and hydrophilic region of the protein, through which it is facilitated to get the substances in. Active transport. What is active transport? The movement of the materials through the membrane against a concentration gradient is called as active gradient, because it is requiring energy from the cells. You can see the plasma membrane in this intrinsic protein, the carrier proteins which can take the particles from outside to inside and inside to outside. So the cellular energy is needed, ATP is needed for doing this active transport, because it moves the substances against the concentration gradient. So passive transport are of many types. That is, one is simple diffusion. That is, simply they diffuse the non-polar hydrophobic molecules like lipids from high concentration to low concentration gradient. They will move very easily without any facility made by any protein molecules. Facilitated transport means diffusion of polar hydrophilic molecules through a protein channel. So that is from high concentration to low concentration, but through a protein channel, so that it is called as facilitated transport. Active transport means diffusion against the concentration gradient, because from low concentration to high concentration it moves, so that it uses a protein pump. For that function, it requires the ATP, that is energy rich molecule. This is the transport summary. You can see in the diagram. Simple diffusion, that is in the plasma membrane without any proteins involved. There is diffusion of substances. That is what called a simple diffusion. Facilitated diffusion by some kind of proteins. They will facilitate. So these two are called as passive transport. Simple diffusion and facilitated diffusion, both are called as passive transport. Active transport, in which it requires the energy molecule like ATP, so that only it can move from low concentration to higher concentration. What about the larger molecule? So moving larger molecules into and out of the cells can be through vesicles or vacuoles by two methods, that is endocytosis and exocytosis. What is endocytosis? Endocytosis again into two types, phagocytosis and pinocytosis. Phagocytosis means solid particles that can be engulfed by the cell, otherwise called as cellular eating. Pinocytosis means cellular drinking. Uh, um, it allows the water to get inside by the process known as cellular drinking. What is exocytosis? Exocytosis means so large particles can be engulfed by endocytosis. It is a process by which a cell surrounds and takes in materials from its environment. You can see in this picture, the waste particles can be exerted out through exocytosis, and the required substances can be taken in or eaten by the cell by the method called as endocytosis. Transport of large particles from, that is exocytosis. Exocytosis is an expulsion or secretion of the materials from the cells. The unwanted waste materials can be exerted out through the process known as exocytosis. Thank you.